continue, right?
A very good morning, good afternoon, and good evening to one and all present here. I welcome you all to the fourth lecture of our COE lecture series on molecular materials and functions. Today's speaker is Professor Robert L. Wheaton, a renowned scientist in the field of chemical physics of metal clusters and nanocrystals. Professor Wheaton completed his BS in chemistry and mathematics from Weber State University, Utah in 1980. He completed his MS and PhD in Cornell University, New York, and continued his research as a postdoctoral fellow at Exxon Research and Engineering Company. He served as assistant, associate, and a full professor in Department of Chemistry and Biochemistry in UCLA till 1994. He was then appointed as Professor of Chemistry and Physics in Georgia Institute of Technology from 1993 till 2013. He also served as visiting professor of material physics in University of Texas, San Antonio, and then served as professor of chemical physics in the same university till 2019. He is currently a professor of chemical physics in Northern Arizona University. His research interests include protected metallic clusters in molecular forms, molecular metallurgy, gas phase metallic and ionic clusters in the gas phase, mass spectrometry, solid state chemistry and physics of molecular crystals formed from metallic clusters, among others. He has over 300 plus publications with 41,000 plus citations and a H index of 92. He has received many awards, including National Science Foundation Pre-Doctoral Fellowship, Nobel Laureate Signature Award for Graduate Education in 1985, Presidential Young Investigator Award in 1987 by National Science Foundation, Sloan Foundation Fellowship and Packard Foundation Fellowship in 1988, Alexander von Humboldt Senior Scientist Award in 1993, Advanced Distinguished Lecturer from Kansas State University in 2008, among many others. Today, he will be delivering a lecture titled Electrospray Gives Flight to Molecular Golden Dolphins. Let us all give a warm welcome to Professor Robert Wheaton.
All right. May I share my screen?
Yes, sir.
Thank you for the kind introduction. Thank you to you all for being patient with me while I try to find the share screen. There it is. Okay, we'll see if it works. I was just explaining to our host, Professor Pradeep, and to anyone else listening that we've just now finished our semester in the past 12 hours or 14 hours maybe now. And so that's an exciting time for all of our graduates and ending this year that I believe, Professor Pradeep, how did you say it? Our COVID children or COVID?
Children of COVID.
Children of COVID. That's a nice, elegant way to say it. You can always count on that. Whoops. That's not a good place to start. Let's try starting with the title slide. How would that be? It's jumping where it wants. Okay, so you've already heard the short version of the title, and the longer version had the word wings, ion catching wings. That'll be a key addition to that. So you've already heard this introduction. I also want to give credit to my close colleague, my former doctoral student and postdoctoral student who's now on the staff here, Dr. Mojamal Haq. He was here until a couple of hours ago, but this being Ramadan, he had to go and get his breakfast or his dinner, or what they call the pre-dawn meal. So I'm alone for at least the start right here. I also want to mention that that this is somewhat of a sequel to some of the talks that you've heard already in this series, and also to a presentation that I made almost exactly, I think it was exactly 6 months ago in Farouk, the Frontiers in Chemistry session, although it's what I'm going to show is all new work since then, or at least work that we couldn't present at that time there. And finally, I want to mention that 5 years ago, almost exactly 5 years ago, I believe it was May of 2017, I was honored by the chance to visit in IIT Madras and had a great week at the low point of the tourist season. So I had the whole coast, we had the whole coast to ourselves there. And I learned a lot on that visit and was also able to present a precursor to this work. And so whether the work is good or elegant or complete, we can at least be credited for, Pradeep, are you listening? We can be credited for persistence.
Of course,
endurance or persistence. So I'm going to give given the circumstances, I'm going to give a very short talk that would leave potentially a lot of time for discussion. And the aim, in case you want a long-term aim for this, is we want to work toward the, I forgot how to use the pointer, but maybe you can see the cursor, point toward the megadalton virus-like clusters. All right, so megadalton is 1 million atomic mass units. All right, let's see, how do we advance? Do I just click on it? Apparently so. So that's the title, Electrospray gives flight to molecular golden dolphins, etc. The words gives flight to, I realized after giving the title that that's somewhat of a metaphor. It means causes to take off, give flight to, causes to take off. And it has both a literal meaning, literal flight, but it also has the meaning of get something started toward a project or a goal. And so I like that. Probably it would have been more literally better to call it lends wing to, electrospray, how electrospray lends wings to whatever these things are, molecular golden dolphins. And if we go over to the left side, and again, I'm not sure if you're able to see the cursor. Let me see if I can get my laser pointer. There it is. Okay. Whoops, that was a pen. Damn it. Okay. How to get out of that. Okay. Try again. One more time. Laser pointer. Sorry, because we're all in person again this year. We lose our Zoom skills quickly, so I apologize for that. So this is a cartoon that just shows how old this idea is. You can see from the caption down below, or you can read it up there at the top. Patrick Arpino is the artist who brings the incompatibility of LC is liquid chromatography, and so a solution phase source, and mass spectrometry brings the incompatibility of these to life. I think the life is also a pun, meaning to biology or to biological problems. And you can see this is from 1982, and it was reprinted in the recent Nature Methods milestone issue of 2015. And so here we have the classic image of a fish who, looking up through the surface of the solution, the aqueous solution or its home base, sees a bird and falls in love with the bird and wishes they could have a home together. But unfortunately, they are incompatible in the sense that they live in different phases. The fish needs the aqueous solution. The winged bird spends most of its time in the atmosphere or on the surface of dry land. Well, there's waterfowl, but that doesn't look like one. And so that's the incompatibility, the fact that they can't find a common home to establish. So that's a common image or a common metaphor, common reference to incompatible lost, star-crossed lovers in the sense of Romeo and Juliet or so. And it occurred to me in using this metaphor that we need the word winged in front of bird. Birds always have wings. Well, the ones that take flight, avian birds have wings. And that's what I wanted to emphasize with this lens wing to here. And then also, if we're going to go all the way into a mass spectrometer, nearly every practical mass spectrometer operates essentially in vacuum, and so that would need a whole nother layer outside of the drag forces of the atmosphere. But you can probably see where I'm going with that. So a more common reference post 1982 is the one that John Fenn at Yale used in his Nobel lecture of, I believe it was 2002. It was about making molecular elephants fly. That's not the exact wording, it's somewhere in here. But in this milestone issue, again, of the nature methods, they reversed the wordings and said, electrospray makes molecular elephants fly. And very nice brief summary, including some key points from that old era. So now we're not trying to make biomolecular elephants fly, but maybe we could call them metallurgical. So we avoid saying metallic, if that's too loaded a term, but metal heavy elephants fly. But going back to the aqueous metaphor, it seemed more appropriate to have a fish. Unfortunately, fishes come in all sizes, and so we're thinking of the really large ones. It would be wonderful if we could, and maybe in a year or so, we can call this molecular golden whales, monstros, you know, the giant mammalian species, non-fishes of the sea. But for now, we're probably still in the dolphin stage. So I hope you like this little warm up and don't find it too fanciful or irrelevant. The key word in all of this was incompatibility of LC and MS, and then how it was solved, at least for the case of small molecules of all varieties and certain kinds of macromolecules, such as biomolecules. So the wings that we are going to be that we're going to be using today, and I don't want anyone to get the idea that this is an exclusive choice, but they are what's known as PEGs, or polyethylene glycols. Poly is kind of an exaggeration. It should probably be called oligoethylene glycol, because, for example, all right, and there's only 4 units of that, and then that's a thiol on that end, so that gives the noble metals have a strong affinity, and vice versa for the sulfur. And then it's very important, as in the case of all the pegylated drugs that you hear about, including the major COVID-19 vaccines, they're all called MPEGs. The little M in front refers to the methyl termination at the end. And we think that maybe made a difference in our earlier failures compared to current progress in that. So let's see if I can advance. Someone will tell me signal me somehow if there's a. Okay, so the Electrospray Ion Source, this is a few centimeters here from the Electrospray needle, okay, the one electrode there, and the spray can be seen by the scattering of light. And here's the conical plate, which is skimmer, looks like to me, conical with a hole at the bottom. And this is charged to several kilovolts with respect to nominal ground being here. And there's 2 important flows. There's the liquid flow that turns with this high potential difference, which turns into a fine spray, and then an ultrafine spray, and finally almost molecularly fine spray. And something has to carry out the solvent. So there's a counterflow of gas, typically a couple of bars, a couple of atmospheres pressure, flowing opposite this. But the fact that the visible part of the flow is still continuing to be bent by the electric field rather than following the aerodynamic flow of the background gas here is a sign that it's charged, and that it's charged in the right sense for the electrode. So, for example, if this is minus 5 kilovolts or 8 kilovolts, then it would be attracting positively charged droplets. And to complete the electrical circuit, it must be that negative ions are are produced either electrochemically or just stripped as electrolyte from the solution, and they go back through the circuit in the other direction. So that's a conventional picture of the electrospray source. And I want to tell you, I'm simplifying and oversimplifying, and to an extreme, just to for the students and more general people in the audience. So this is Fenn. We knew him as Old John Fenn back in the early 80s, and he went on for another 30 years, as you can see. So the chemist who enabled mass spectrometry to weigh up biology. And this is from Nature. This is the obituary written by Carol Robinson of Oxford University, who was one of the great proteges of Professor Fenn. He encouraged many people, Carol Robinson being a extremely notable example in the what's called native electrospray mass spectrometry, intact ribosomes and viruses measured in high vacuum to several megadaltons, there's the megadaltons, and even deposited and recovered live in the sense of not deactivated from their experience as the proverbial fish out of water and even into vacuum and then back onto a solid substrate and recovered in solution, reconstituted or at least retaining its enough of its structure to be active. So that's a famous part of modern, I would say, chemical physics, but molecular biophysics and other areas enabled by mass spectrometry. And the only word we would like to change, those of us who are working on this frontier, is we would just like that word biology to be replaced with metallurgy. Metallurgical molecules. Okay, let's see how I'm doing on time. All right, so this is what it looks like, what to expect from a modest size, on the small size, globular protein. For example, lysozyme was one of the first ones that was ever analyzed in this way. And it has about 14 kilodaltons. Of course, lysozyme is essentially an organic molecule. And I want to focus on the organic content of the gold cluster masses that we have here. So we think that's probably the easiest fair comparison, is to talk just about the organic part of the mass when it comes to what to expect in terms of solution phase and electrospray properties. At least that's the aim. So this is a useful little paper by Douglas and Konerman from British Columbia. It's 1998. I see I didn't write the date here. And their focus on this was to finally establish what was already suspected, which is that proteins are in danger of unfolding. So, a globular protein has a three-dimensional globular shape, approximately spherical on the average, and yet in strong acid, base, temperature, etc., too much salt, etc., the proteins will denature, and technically that's referred to as the unfolding, because the folded form is the compact form. And so they distinguished this, and they did it for three, in this paper for three: lysozyme, cytochrome C, and ubiquitin. And so, for example, here's the charge distribution one gets from electrospray. Positive ion mode and negative ion mode is chopped off there. When it's a strongly acidic solution, say pH 2. And this is buffered to be near pH 7. And you can see that in this case, there's a very broad distribution, and one can get quite high charge states, always remembering that a mass spectrum is the abundance of ions plotted versus the mass to charge ratio. The charge represented by the letter Z or Z here is an integer multiple of the fundamental charge, the electron charge, or the positive version of the electron charge, right? So that's what these integers represent, and the positive indicates they're in positive ion mode. So there's this broad distribution, and again, because the charge is in the denominator, then they become closer and closer together, and that's expected. But it's all believed to be the same lysosome, because the masses are related in this algebraic way. In the non-reduced state, there's only 2 or 3 charge states represented, There could be a little bit of the denatured, but it's not really showing up. And it terminates quite strongly at 10 plus. So we might want to remember that 10 plus goes with 14 kilodaltons, or I'll try to remind you with that. So I hope that's clear what we're talking about here. Lysozyme, right? Like from egg white, protein, Eiweiß, right? Auf Deutsch. Here is ubiquitin, the kiss of death, small protein, 6 kilodaltons, so-called because when a protein gets ubiquitinated, gets attacked by ubiquitin, that marks it as damaged, and the cell can go ahead and destroy it, metabolize it back into its bits and pieces, amino acids, to be removed, broken down further and removed, right? And so this is only 6 kilodaltons, very small, only like 50 odd amino acid residues. And it's the same situation. If the pH is too low or too high, you get the broad distribution. And if it's buffered in this nice zone, 7.2, like physiological conditions, it peaks strongly at what looks like a maximum. You still see some of the others, but those were interpreted as being, even under those conditions, the transition, the temperatures, pressures, whatever, of going into vacuum are considered as possibly producing the denatured there. And this is the negative ion mode, which I won't have more to say about. All right, so that's 6 kilodaltons, about 6 plus, okay? And here's just an intermediate one, about 12 kilodaltons, not 14 like the first or 6, but about 12. And here it's peaking around 8, and maybe there's 9 too. So these are the kinds of mass spectra that can be observed for intact and attributed to native, meaning native folded structure in the gas phase. And of course, many other things have been done since then, like mobility measurements and so on. But I'm just restricting it now to the mass measurements. So now here's what it looks like moving away from biomolecules, molecular biophysics or biochemistry, if you want, molecular biology, and moving over to the clusters. So the first one we want to look at is just an old favorite, Gold 144. And we're going to start with all benzyl thiol. That's benzyl is CH2 and then a phenyl group. So it's by composition, it's C7H7, okay? There's a crystal structure for that with all 60, and it's very nice, beautiful, and all that. But we're starting with that, and then we're doing an exchange reaction, okay? So we're replacing many of these ligands. This is all around 65 degrees C, and I can give more details for anyone who wants it, the solvents, the phases, and so on. So down at the bottom, we have a small piece of the mass spectrum. I hope you can read it. Again, it's mass to charge ratio on the base, and it's from about 4,000 to about 10,000 daltons per charge, daltons per fundamental charge. And so these have been assigned previously. I should say these samples, very pure, came from Flavio Moron's lab. His postdoc Tiziano Dainese produced these. And the highest charge state we could see was 4 plus, and it really is most strongly peaked at 2 plus, which is way off the scale here. So if we accept that old assignment for this, and we want to know what the total mass is, we would take that 9,000, approximately 9,000 there, multiply it by 4, that's about 36,000, or in the conventional language for this macromolecular mass spectrometry, would just say 36 kilodaltons, understood. And then when it's reacted for 9 hours, say, sometimes you're going to see some down to 1 hour with the MPEG-4SH. This is the famous thiol-thiolate exchange reaction known for self-assembled monolayers on surfaces for 35 or 40 years, and on clusters or nanoparticles for a good 25 years. It's a very facile reaction, handing off the proton to the outgoing thiol. And now you can see this whole suite of peaks. Now, because the MPEG-4 here, thiolate, is quite a bit heavier than each exchange gives you a peak at a higher mass. So this, roughly binomial shaped distribution is just like 2 exchanges, 2, 3, 4, 5, 6, 7, 8, 9, so on, like that at that point. And then if you go on another 10 hours, say, you push it even farther and you're getting up toward 15 and 20 and so exchanges. And if you elevate the temperature a bit, now you're all in one phase, which I'll describe the phases in a moment. But and you get up toward a day and a half or 2 days, the reaction can be driven essentially to completion. And now the charge state is, well, 4+ is missing. Okay, it would be over here, but here's the 5+, 6+, 7+, 8+. And Dr. Hawk insists that this fits very well to the 9+, although with this PowerPoint slides, it looks like it's in the digitization noise of the thing. So this, maybe it's not quite complete. There's some other peaks that would indicate, 59 exchanges. So one of these remaining, 59 here, and 58 and so on. And there's other satellite peaks. It's not as clean as we started with, but it seems to be pushing toward completion, and probably 8+ is about the highest charge state that we can get out of that. Let's go back for just a minute. Where was 8+? There it is. Cytochrome C with about 12 kilodaltons of organic is topping off at 8 and 9+, if we rely on this old paper from the literature. And here we're also, looks like we're tending toward 8 and 9+, depending on how the instrument is tuned and everything. That's what he thinks for these preliminary kind of results anyway. And now you might ask, well, what is the volume or the radius comparison here? If there's electrostatic energy is a big part of how much these things can be charged, then, and so we can do that. We can go 8+ times, assuming this is completely exchanged. Now we're gonna just calculate the total mass and then subtract the core mass of the gold. So 8+ times 5,000 is about 42 kilodaltons. Part of that you're going to see is some small ions that are caught by these wings of the bird, right? But it's a small part, so we could take 41 or 42 kilodaltons. The gold 144 is about 28, a little over 28, 28.3, 28.4. So taking the difference, 41 and change versus 28 and change, it's about 13 kilodaltons of organic mass. 13 kilodaltons versus 12 kilodaltons. So it looks like maybe the same principles that applied in the biological macromolecule mass spectrometry as represented by the protein cytochrome C may be operating in the case of these quite artificial seeming pegylated thiolates of the same total organic content. So we get a nice large charge enhancement, multiple charging, that could be useful in making this generally applicable on many other instruments. So now, given that example, and by the way, I mean, I could just, if I'm allowed to escape from this, presentation, I could just show you real quickly a few other examples. Now, I'm not pretending, unless you're quite experienced with electrospray mass spectrometry, you're probably not going to follow this, but let's just pretend for a moment that some of you in the audience are very well practiced with this, at least for smaller mass species. Maybe some of your organic colleagues or biochemical colleagues have a compact version of this instrument, and the only thing that's then would look unusual to them is that given the charge states like 8+, it's quite unusual that we're still up around 2,000 mass to charge, which just means we're in the macromolecular regime as far as they're concerned, polymers and biopolymers, and not in the molecular regime. So this is an example here, and it's awfully compressed, but that's the way they present their things, and I apologize for that. So on the top here we have retention time, okay? And so this is retention time of a micro scale, analytical scale, HPLC column. I'm not going to talk about the mobile and stationary phases and things unless we need to. And this is locking in on various mass signals. Unfortunately, there's not a total ion chromatogram in this scale, but you can imagine summing these things all up and getting a broad peak. And this is just a horrible mixture. You can break it down to all these components, but you could probably even get more components. And the reason it is that is because it's at the midpoint or near the midpoint of one of these exchange processes. Now this happens to be, it doesn't show it here, does it? But maybe here. This happens to be Glutathione Gold 25, which has been popular in several of the recent talks from Japan and Singapore, Professor Tsukuda and Shia. And Gold 25 started out with 18 glutathiones, which itself is quite a massive thing. But this is MPEG 12, which is even more massive. This is about 575 Daltons, and the glutathione's about 305 or so. So, each exchange, it gains about 270, a quarter of a kilodalton. And if you're going to exchange all 18, it's going to gain a lot in mass, right? And so this is quite a lot of excess. Oh, we lost the pointer, I'm sorry. I'm doing all kinds of signaling, but this is quite a large excess of now I'll point again to it, the incoming ligand versus the outgoing ligand. And maybe a very boring kind of chemistry, this exchange, because you just get kind of entropy driven or statistical distributions. But anyway, that's the pattern. And these are quite a bit longer, okay? So even though the core is small and there's only 18 versus 60 in the previous examples, it appears that we're getting all the way up to 10 plus. I'll show you the isotopic evidence for that here in a few nanoseconds. So let's go back to this example. And now if we look at so-called fraction 1, this first peak in the chromatogram, all we have are typically 2 exchanges, okay? Interesting that 2 is very weak here, so it's really 3. Remember, we gain mass. I'm sorry there isn't a reference spectrum of the gold glutathione on this trace, although I have to tell you, normally you have to switch to negative ion mode from positive ion mode to do the gold glutathione. It takes heroic efforts at adduction to get into positive ion mode with gold glutathione clusters. Anyway, so, and this is a prominent peak, that's 2 exchanges, and that's from that first sample. And the charge is identified from the high resolution isotope pattern to be 4 plus. And this is, okay. I hope I'm saying everything right here. He coached me on it a few hours ago. And then the second peak, if we take this promontory here to be the second peak, now these are the lines here indicate where the 3 exchanges are, okay? Is that right? Yeah, 3 exchanges, and that's 4 plus, 5 plus, and 6 plus. And so you see a very steady pattern that as you move to longer retention time on the column, you're getting things which have more exchanges, like 2, 3, 4, 5, 6, 7 in this simple pattern. And also the charge state, the intensity distribution is shifting from low charge states, low positive charge states, to much higher ones like that. And in fact, when we look at this in detail, we find the most marvelous little correlation that every time a new charge state appears, it coincides with 1 extra exchange. Now, I said you wouldn't follow this unless you're practiced at looking at these. And it doesn't help that we have all these integers to represent the different fractions, integers to represent the different number of exchanges, integers to represent the different charge states that have been assigned. And so it's pretty much hopeless, but that was part of my point. It's a specialized business. If we take a look at the isotopic patterns, like let's take a modest example first, 5 plus. We can fit those very nicely, the isotopic pattern. And it's interesting that it requires to get the 5 plus state. Well, this has complete exchange, sorry. So you're not going to learn about that threshold arithmetic that I just explained. But it is interesting that to get the 5+ charge state, there are 6 ammonium cations worth of mass, 6 times 18, 18 being the mass, 18 Daltons for each of these. And okay, and we think that's significant because the gold 25 thiolate 18 likes to be -1, -1 in the charge state, and adding 6 cations, whether they be sodium ions or any other ions, but in our case, we're using ammonium formate, ammonium acetate buffers. We assign these as ammonium. So that's necessary to get you can see it's a high resolution scan. Important is that each one of these peaks really are 1 Dalton spacing in the charge state 5+. So that means they're 1/5 or 0.20 in the unit Dalton per charge, which is sometimes called the Thomson of unit when you take a mass and you take the ratio of it to the charge, fundamental charge. So this suffices to prove that it's charge state 5. Pretty good confidence that that is dominated with the 6 ammoniums and that that's consistent with a core charge of -1. So no surprises there. And indeed, I hope that nothing I show will seem surprising. Here's 10+, okay? We could, if you wanted, go back and point to that. Why don't we just take a nanosecond to do that? 10+. So this was a pretty iffy thing, especially because this is not at all the thing we're talking about. It's down here. 0 remaining glutathiones. This is actually something totally different. A singly, or maybe, sorry, it's a doubly charged species. So that's you know, an oligomeric gold thiolate in the solution. But anyway, 10+, and so now the spacing between the peaks is 0.1 in the unit Dalton's atomic mass units, that is per charge. So, you know, they say if you've seen one of these, maybe you've seen enough, you've seen them all. But I want to mention just one other case. We use this DMAET. It's a very very nice ligand. It's the basis of Dr. Hawk's thesis work in the University of Texas. And it itself is an aminoethane thiolate. It has a common name, captamino, okay? Captures, it's an amine that's a thiolate on the other end, and it's small and it captures heavy ions. So captamino thiolate. And it's also another easy way to look at it is thiocholine. Choline, like acetylcholine, is a major role in neurotransmitters and all that. So this is just the thiol instead of the hydroxy version of choline. Sorry, there's no structure on this slide. But you know, this is something that he did. Here we have the starting material, and again, because this has an amine, a dimethylamine terminus, it captures charge, or at least protons, pretty well. So it'll protonate. It's 6.8 kilodaltons with gold 25. So doubly charged, it's 6.8. It's 3,400 in mass to charge ratio, and we can get the triply charged, but nothing we can do will get us to the other higher charge. When we put the wings on this, the ion catching wings, then with just a few, just you know, enough to hold 1 ammonium, I guess, you get 3+ and 4+, and the 2+ is gone. And then I think maybe this is as far as he took this particular case, PEG 6. Yeah, okay, so PEG 8, the reaction isn't complete. That's why you have so many different things, but you're already up to 6+. And we think 6+, you know, maybe, you know, there's a symmetry to this thing, why it might be quite happy having the 6 charges. If you know if you remember the molecular structure with the 6 long staples there, then there might be a reason to top out at 6, quite apart from the electrostatic limit on that. So I think maybe, you know, that's enough. Of course, you can make very beautiful diagrams of these showing the different retention time of the starting and isotopic high resolution to demonstrate the assignments of the charges. And then you get up to this. Here he has Z+1 ammoniums for that, and the red is the retention on this. So let's see. I think we already looked at this. I think that's probably enough, although it's hard to stop because, you know, it was fun work for him. So I just talked about this now in such a way that, that again, unless you're quite used to looking at it, you can probably hardly follow the logic. But now I want to finish up by talking about, by talking about things that are not obvious in this. Okay, and so let's remember mass spectrometry basics. Okay, Graham Cooks, we heard him say, the least interesting part of matter and mass spectrometry is the mass spectrometer itself. Instead, we have all the interesting chemistry in the ion source, and then all the other non-mass measurements. In other words, things I didn't talk about at all, like ion mobility and deposition and electron diffraction, like Manfred, I hope, will tell us about the latest in that. All the other things you can do with the ions once you have them in vacuo or back on a surface or something like that. And so we've used all of this along the way. And now I want to go into the secrets, such as they are, for the ion source. And the question you ought to have in mind, or at least the question that I agonize over for the last quarter century, is why couldn't we do this in 1996 or 98 or 2000 or 2004 or so on, okay? And so I'm going to skip this about standards. We can come back to that and about isotopic resolution at 36 kilodaltons. And let's instead we'll skip the time of flight mass spectrometry and custom instruments. And let's just ask the question, focus on this question for the remaining time. What is wrong with LC-MS, liquid chromatography, mass spectrometry, and all that would be enabled by that, if it would only work, of the metallic macromolecules? And in 2016 in Davos, the great S3C conference there, we posed that question, and the answer was everything. And I gave example after example in the end of a talk where everything we did out of non-aqueous, non-methanol, non-biological type solution seemed to be working pretty well, but everything failed going toward the traditional conditions of macromolecular ESI. And again, 14 months later, during my week-long visit to Chennai, the same answer. And so everything means the answer seems to be to remember that proper macromolecular ESI conditions, in other words, the kind you would find your colleagues in molecular biology working with all the time for their work, include all of these or achieve all of these limits. One is they work from a standard set of solution phases. They hate to go away from these solvents or mixtures of these solvents. Water, that's our fish out of water, aqueous solution. Methanol, acetonitrile. They'll do it in desperation, but they hate it and they hate to report it, and they won't listen to us normally. Not that we try to talk to them, but get their criticism, but they won't talk to us about any of our problems, any results, unless we're in those phases, okay? And that applies also to the mobile phases, so-called mobile phases in the chromatography. Number two, the charge distributions are pushed towards some kind of electrostatic maximum, where they don't seem to be that way in the case of getting metallic macromolecules out of organic, non-polar or less polar organic solvents. Like we really like to work with dichloromethane, DCM, because it's so volatile and it's non-flammable, et cetera, et cetera, et cetera. No, you can't even you can't even get advice from the mass spectrometry people if you're using that. just speaking from experience now. A third thing is they're typically working with sub-picomole samples. And I mean the sample that would need to be injected in order to get a good high resolution spectrum and resolving many, many components. So please don't think that one component or one charge state of one component accounts for the under 10 nanograms. No, it's probably more like, half a nanogram, 500 picograms or something of material. And so we're talking about working with less than 10 to the 12th molecules, chargeable molecules. Another feature is they have very fine control over the amount of electrolyte they use. Everything in their work is very quantitative as compared to if you read any of the papers in our field of metallic clusters, monolayer protected clusters, you'll find that it's very vague on this kind of information. Also, they work in a way that lends itself to automation. Not only the running of the whole LC system and data collection, but they'll use auto samplers. They won't trust manual or just direct infusion. And they're down to around 1 microliter on this auto sampling, okay? And finally, you already saw pH mentioned, electrochemical potential, temperature, all of these things are under much tighter control like this. Now, I want to tell you just briefly what some of the responses were. And most of these came out of this Davos meeting, because up until then, we had never exposed our struggle. And, when I say struggle, I'm not trying to make anyone feel sorry for us. We were able to publish and help our colleagues, especially in the biophysical side. It's just that the results were miserable and they were hell to obtain, required torturing the instrument and the samples in every way. And Professor Tsukudison could probably elaborate more because he's worked with them quite a bit. So here's some responses that we got. MT Bowers, I think many of you know Michael Bowers, very illustrious Santa Barbara, California guy, editor of JAX, editor of whatever. He responded to our talk in Davos, and especially this part, and said, please tell me that you are using nanospray, not electrospray, conventional electrospray, which is what I referred to here, but nanospray. Nanospray is typically defined as well below 1 microliter per minute of flow rate, and it's femtomoles of sample. That's what he found worth commenting on in the work. And this points out something that I cannot emphasize enough to people working on the gold clusters, gold, silver, gold, copper, etc. The gold, the metal, is the cheapest part of the experiment in macromolecular biology. Everything they work with is so much more expensive that they benefit greatly by going to extremes of low sample size, low flow rate in order to complete all their experiments. That is, by the way, the reason why there's a possible chance that the gold clusters could become standards in this high mass and ultimately toward a megadalton range. It's not only because they're very isotopically clean, but they're being mostly gold when you get up into that mass range, but also because they're very inexpensive. You can regard them as synthetic or as semi-natural products, but they're much cheaper than any kind of biological, pure macromolecular biological sample in that range. Graham Cooks, when he visited us in Texas, San Antonio, he said, please tell me that you're fabricating the clusters in an inline reactor. And I had to tell him, no, when we say inline or online, all we really mean is that everything's connected. There's no break between the LC instrument. I mean, there can be all kinds of optical detectors and things, electrochemical detector in the capillary flow, but there's no reactor going on. Philippe Dugord, what he got from Lyon, what he got from this was, wow, we're using Orbitrap for this. And, since those are, as Pradeep will tell you, those are very expensive and specialized grade instruments, he was impressed with that. When in fact, all we did was use the old Finnegan demo lab in San Jose, California for the very high resolution work, high temperature, electrospray source, all of that stuff. Pradeep had a good comment. What's the use of doing nanogram separations? Well, and he's right. They're useless in terms of producing a material that you can use for other bulk experiments. They're useful for deposition, but things like that. And speaking of deposition, Julia Laskin in 2019, when she started to work on these gold 144 clusters, the same ones that we were using from Italy, she was the only one who would say, don't worry about all these other things from the experts here. You know, you're doing exactly the right thing for this.
Rob, we are at 52 minutes, okay?
Yeah, that's great. That's great because I'm done. Yeah, I'm really done. So I'll stop on that note. Thank you very much for your patient attention and for putting up with all these concerns. Shall I stop sharing screen?
Shall we open it up for questions?
Yes, please.
Very nice, very nice. So let me, very nice, Rob, to see this. When clusters take wings, they take wings like proteins.
They take flight. We lent them wings. Yes, sir.
Yeah. So, and it's those wings are really fascinating. The charges are fascinating, and the behavior tend to be that of proteins, macromolecule is also fascinating. So when people look at proteins, you relate them to point of zero charge. At
Yes.
point of zero charge, of course, no charge, and therefore that is reflected in the mass spectrum. Are you saying that their physical properties, such as pKa, would be similar to that of macromolecules?
Yes. Thank you. Yeah, that's a very good point. And the short answer is yes. At that pKa, that pH, that electrolyte concentration, we are trying to make them as similar as possible without doing all kinds of fancy synthesis. You know, imagine you had to do this with a globular protein, with an enzyme that you wanted to vary the surface charge. You would have to do something that the biologists call site-directed mutagenesis or something like that to vary the number of chargeable groups in the periphery of the protein, the enzyme complex in solution. That's right. But here in this this crazy world of surface modified clusters, we can do a cheap version of that. Of course, we don't really control where those charge sites are, right? It's statistical up into the limit of complete exchange. Is this helpful, or am I drifting?
It is. So, you know, the charge and the pKa or pH or whatever that you may have would tend to suggest additional properties of these systems, their assemblies and organizations.
Yes, sir.
Now, that if one is relating the macromolecular mass spectrum to macromolecular structures, similarly, macromolecular mass spectrum, so cluster mass spectrum to protein-like cluster structures, the secondary structures, the assemblies, the organizations that we may envision, they're all likely to be similar. So building on this will be the next step, and that's a great suggestion that you are making. Now, does it really mean that this kind of a behavior is there only for spherical systems?
Right.
Is there a structure dependence to it? Those are associated questions as well. I stop here. I look forward to your comments and then open it up for additional questions.
Thank you. Let's work on it. Let's work on it together. We need help, all the help we can get.
Sure. Okay, Manfred, you have a question.
Yes, hi Rob, thanks very much for your nice talk. I wanted to ask more about the charges. So I think you implied somewhere in between what you were saying, that if you put these highly charged things into gas phase without a solvent around, then there's a limit, which depends, I guess, on where the charges are relative to the center of these particles, and and also the size. And otherwise they Coulomb explode or something like that if you charge them up further. So is that what you were saying?
Yes. Now I didn't want to say anything like Coulomb explode, which is what the drops are believed to do, but rather they would simply desorb one of the excess ammonium ions, either to solvent or that's the way Encke, Christy Encke and so on tried to resolve the paradoxes of formation. But this question of where are the charges, okay, is really a good one. And let me just give you kind of a historical answer real quick. Fenn's group in Yale, when they were still in Yale, they did polyethylene glycols, a polymeric distribution, because they couldn't purchase or synthesize the pure finite length. And they proposed a model, electrostatic model, for exactly what we're talking about, reversible binding of sodium ions to the PEG. And they imagined it as a long linear polymer, like beads on a necklace, right? The different sides. And they extended it all the way out. And they got answers which were off by a common factor. They were getting charges far too low, but the systematics were in the right direction. Now, since then, and maybe even at that time, organic chemists, physical organic chemists knew that the way polyethers, that's another name for what we're talking about here, polyethers, when they organize to bind an ion in solution, and they can bind it very strongly, like crown ethers, okay? You know, many electron volts, don't ask me for the precise number now, but out of solution to bind these ions, they organize a cavity for it with all the oxygens pointing inward. And for a small ion like lithium, it would be 4. For sodium, it would be 6 ether oxygens. And for the big ones, like actinides or something, it's 8 to 12 are required. So we don't know. You're probably going to say that ion mobility will tell us, and I love that. Well, Wendell Griffith, our partner in San Antonio, told us that with the timsTOF instrument, he'll tell us right away whether these things are extended like a star or they're collapsed on the charges. I don't know. We'll see. But anyway, you understand what we're thinking is that with these short pegs, that it's multiple of them that, with the long ones, a single one can reorganize to form a capture, a captain.
And multiply negatively charged analogs don't exist. Or do they?
Yeah, no.
No, okay.
The lore of pegs is to go positive.
Yeah, okay, thank you.
Yeah, thank you.
Yeah.
Love talking to you again.
Yeah, Tatsuya, you have a question.
Hey, thank you very much for the nice talk, Rob. I have a related question, maybe. So when we cannot crystallize the clusters, the only way to know the charge state of the clusters may be by ESI, mass spectrometry. So my question is it always possible to take the cluster ions intact in the gas phase by ESI? Maybe ESI can ionize the clusters, so then the information about the charge state in solution is lost. So I want to extract the clusters in the intact form while keeping the charge state. Do you think it's possible?
Yes, I do. I don't believe that we can do that, but I do believe that the combination of a good electrochemist and a good analytical electrospray chemist together can do what you can get the information you're asking for.
So they take wings with the charge.
Yes, charged wings. The long version of the title had ion catching wings. Electrospray's ion catching wings.
But you change the charge state as well as retain the charge state. You can do both.
Yeah, that's
And
right.
Tatsuya would like to have that information, retain the charge as well as add charge with control.
Yes, that's right.
Okay, thank you.
I'm tempted to write a little more in the chat line, but I think we should probably I want everyone to think independently and not accept any story, even a simple story that I give, and we'll learn more from you when you bring your own. We argue about it among ourselves, Dr. Alvarez, Dr. Hawk, Dr. Black, Dr. Griffith, myself, 5 people, 5 totally different views on how the charging works, the core charging, the surface charging, and now the ion catching wing charging, the interplay among these. And everyone has a very strong opinion. Of course, I think my opinion is the simplest explanation for what we see, but the others are very persuasive people too, and I expect no less from doctors.
Pallab, you have a question? Pallab's question.
Yes, sir.
Yeah, go ahead.
Wrote a long message in the chat.
Yeah, that was me.
Yeah, go ahead and ask your question.
Yeah, hi Rob. This is very nice talk. Let me read out the question for you. So, as you said that the multiple charge states of the clusters are quite similar to the protein charge states, right? So we know that protein charge state used to change when certain environmental conditions are changed, like if you change the pH of the solution or the temperature of the solution, the charge state change. Do you see the similar thing or similar effect on your exchanges or the charge state of your clusters while some parameters are being changed, like temperature? If you change the temperature, do you see the exchange kinetics being shifted to some other direction, lower charge state, higher charge state?
No, it's a very good question. Very good question. We don't believe that, but we could be wrong. We don't believe that it's possible to denature in the sense of unfold the monolayer protected clusters in the same sense that the proteins would do. And I saw Dr. Kappas shaking his head when I suggested that he was already thinking that ion mobility could resolve fine features of the charged cloud around this. But he would probably say, yeah, if you opened up the whole cluster and extended it to a very elongated, that could be a temperature and pH and et cetera change that could be detected with an advanced mass spectrometer with ion mobility and things. So that's a very good question. Certainly the other question you asked, do you see addition of cations or anions? Yes, cations. And we try to control the cations that go in there. People say, oh, you should just use it as it is, and it'll take sodium ions out of the glass, out of the materials of the instrument. But we don't like that. We would rather use ammonium or something like it and use deuterated ammonium. You want to know that what you're adding, the so-called parameters of the experiment, are things that are within your control. But I can't overemphasize how preliminary, how primitive and preliminary it is, everything we do. The worst mistake I could make in this talk is to somehow confuse you into thinking that we are doing something sophisticated or attempting to do something sophisticated that advances in any way the state of the art of electrospray mass spectrometry for macromolecules and even the coupling to liquid chromatography. We're intentionally doing everything as plain vanilla, garden variety, whatever as we can, in order that we can get help from the multi-billion-dollar investment that's been made into molecular biophysics and bioanalytical chemistry of biomacromolecules. And that's why it disappoints people like, such as I mentioned, Professor Bowers, Professor Cooks, Professor Pradeep, they would rather see us do something more exciting than what we do.
Well, this is quite exciting. I mean, it is. Are there other questions?
You know, a question that Professor Tsukuda and Negishi-san asked when we visited Tokyo for a week back in 2015, they didn't ask it in these words because, you know, they're gentlemen, Japanese, polite, but it basically amounted to, how can anyone be so rich as to have one of these high mass electrospray mass spectrometry systems dedicated to our work, you know, and not being guarded by our colleagues who depend for everything they do on those instruments. And you know, that's a very good question. It's also a question like, why work with gold if you could work with copper or carbon or something else like that? But I think we can answer that. I don't think that these instruments, for what purposes we do, they necessarily need to be that expensive. We rehabilitate first and second generation time of flight mass spectrometers that we can get $10,000 from eBay, you know, that are completely intact. They just have gone out of fashion. What's really expensive is the people, the expertise to keep the instruments clean and running and all that. But that's what chemical physics types are good at, you know, so why?
So Rob, I want to take you to your 1996 paper.
Yes, sir.
Wherein you measured the mass spectra of nanoparticles.
Yeah, I'm here.
So now the question is that if this methodology were to be available and to be used with nanoparticles now, you would, of course, have seen in 1996 that all those nanoparticles are also molecules.
Yes.
Now, what stage are we in seeing nanoparticle mass spectra with this methodology?
Yeah. Yeah. So why am I showing only gold 25, gold 38, silver 29, gold 144 and everything? I want to remind you that even since 1996, the group of Amala Das in Oxford, Mississippi, and his student Chanaka Kumara, they were able to get decent electrospray, even up to 200, even at 300 kilodaltons. They found a series of magic sizes. Okay, now the spectra won't convince a skeptic because the signal's very weak, the resolution's very poor, but maybe I lost sight of your question, but we believe if you have an instrument like an Orbitrap, extended mass range, ultra mass range, that can do the viruses, 3 megadaltons, 1.7, 2.7 megadaltons, that the cluster preparations, etching, annealing, and so on, will ultimately produce samples worthy of that. That's just a belief. It has to be tested.
Well, one has to go to probably charge detection, mass spec, and many others, but the methodology that you presented is very interesting.
Yeah.
So, Lou, you have a question, and then we can probably close this. Yeah.
Charge detection is one way to go, but with the high charge states, those viruses are detected by,
Lou, do you have a question? Yeah, quickly.
Hi, Rob. I have a question. Like, what's the largest cluster measured so far? Perhaps you answered that already, I think. Like you mentioned, 200 kilodalton or so. So, are there any plans to do in-solution mass photometry also on these clusters, which is developed by Philip Kurkura from Oxford? though it may not give high resolution info like ESAMS.
Okay, what is
and
the
also
could you repeat what was the method you mentioned?
in solution mass photometry.
Yeah.
Mass
Mass
photometry.
photometry. Yeah.
Okay, I'm afraid I don't know the.
It works based on the scattering.
Yeah, well,
Scattering interface.
similar to
Scattering
DLS, but Lure that doesn't have that kind of resolution.
yes, that's what I mentioned. Yeah, perhaps for your really, and also for really large particles, there will be plasmonic effects that will affect the information what we get. But in this case, which where you're using a lot of PEG and really small one, which doesn't have plasmonic property, maybe that it may be useful to characterize the in solution mass before going to the gas phase. That's what I thought.
Great idea. Yeah, we should do that. It should be done. Maybe you'll help us do that.
Vicky Waisaki is doing this, Rob, and so maybe she may be interested in doing this. Yeah.
We just saw Vicky in San Diego at the ACS meeting, and I hope I'll see some of you in Minneapolis, the ASMS meeting, mass spectrometry meeting, annual meeting. The first full week of June is all in person again. So.
Subarna Maiti, with that we will close. Subarna, you go ahead.
Yes, sir. Nice lecture, Professor Whetten. I just have a simple query that I have come across the fact that even inherently negative charged gold nanocluster also gives signal in positive source ESIMS. So that's why we sometimes got confused that is it originally positive charged or it is giving the positive signals due to the ion source. So how can we just face the problem, solve the problem? Is there any straightforward way, or it's just the modification of ionization sources and the mass recording condition?
So this is about changing the sign of the charge.
No, the inherent charge, how do you measure? The same concern as Tatsuya was talking about.
Say it again, I'm sorry.
The inherent charge, how do you measure that? Because ionization changes charge state.
Yeah, so we don't have any direct way to measure it, but let me try to explain something very simple. I call it Mohs law because his name is very short and he found it. Okay, so let's say that we know gold 144 really prefers to be in the 2+ charge state. It takes enormous effort to see the 3+ and 4+. Of course, I'm exaggerating somewhat, but let's say it prefers to be 2+. And even under strong acidic conditions and so on, and tuning the instrument, it's a big fall off to 3+. Let's assume that 2+ is the preferred state. And now the ligands are not the kind of ligands that can capture protons, can capture charge in any way. So let's say you know that independently. Then what's interesting is if you take a long peg, assume the pegs are independent on there, you take a long peg like peg 12, and each time you add one more peg, so say the first one, that peak in the distribution shifts from 2+ to 3+. Now you add one more peg, okay? You don't actually do it in sequence, but in the detector, that's how you see it. You have now only 2 pegs. Now you get to see the 4+ as the most prominent of all the things. Do you see what I'm getting at here? It's as though the charges up to some limit are independent, the core charge and the peripheral charge, like that.
Thank you. Thank you, Rob.
That's a nice
We're running
way.
out of time, but
Thank you, Professor.
Thank you.
I'll send you the arithmetic of the Mohs law.
Thank you. Well, giving wings to these golden clusters, golden molecules, would probably be a way to study nanoparticles and their molecular character in the years to come. And maybe that is what we will do. Well, this understanding will give us greater insights to organization and assembly of such systems in the foreseeable future. Thank you, Professor Rob Whetten, for that wonderful lecture. So we will meet next month with another exciting lecture. Till then, goodbye.
Thank you. Goodbye. Good night.
Thank you. Bye.
Thank you, Thomas.
Thank you.